Okay, so we are going to attempt to audio record some of the lectures. This is Chapter 7, Part 2, what feels like about take 99. So we are going to pick up where we left off before spring break, which is in part of Chapter 7. So where we left off in Chapter 7 was with the neutralist selectionist debate. At the heart of this debate is the question, which is more prevalent? Neutral evolutionary change, i.e. neutral mutations are occurring more commonly, or selective evolutionary change, which would mean that we would have advantageous mutations that are then acted upon by selection. So which one do we think is happening more in natural populations? So we often know that there's many processes that occur at the same time, not just a single one. So you don't have a population that just has mutation or just has drift. It's usually a multiple mechanism. So what we're looking at here is what happens if we have, let's say, these three primary mechanisms that are out there: mutation, drift, and selection. And they're all working together. What's going to be the overall outcome? So there's three possible outcomes that they look at. You can have harmful mutations which would arise, and then those would get eliminated by selection, right? Which we expect for a harmful mutation. Secondly, we might get a neutral mutation that would arise that would then go to fixation or loss, probably 50-50, depending on what it started at in frequency. But if it's neutral, it could be one or the other. And then we also could see advantageous, beneficial alleles popping up and then becoming fixed in the population because selection favored them. So those are three possible things that could happen. Now the first one of these things we're just going to eliminate. And we're going to ignore it because, well, those alleles that went through that are eliminated and they're not here today. We don't see them. They get taken out pretty quickly. So we're going to just ignore those things and focus on 2 and 3. And when we focus on 2 and 3, what we're going to really be asking is, which one do we think is happening more, or are they even? So whether we are going to abide by the neutralist theory or the selectionist theory really depends on how important we think those two things are. If number 2 is more common and more prevalent, then that is consistent with the neutral theory because they're neutral mutations. If number 3 is more likely, then, or more common, then that is something that would be supportive of the selection as part of the debate, because that is saying that you do have these selective forces operating on these beneficial alleles. So this is sort of the heart of those two things, is whether or not those mutations are neutral or beneficial. All right, so let's take a look at what happens to a beneficial allele. I'll come back to neutral. Let's look at what happens to a beneficial allele. You have a huge population, okay, infinite in size. So what happens to a beneficial allele in an infant population is it goes to fixation. So if you go back and you think about some of the graphs that we did from Chapter 6, even if you had a really slight advantage, that allele that had the slight advantage became fixed. It might have taken it longer if it was a really weak advantage, and it might have happened fast if it was a strong advantage. But no matter what, it went to fixation. Okay. Now think about what happened a little bit later in that chapter, Chapter 6, when we went in and we made the population size small. And remember what happened when we basically shrunk the size of the population. Instead of giving those smooth curves that we were seeing, the curve started getting bouncy, right? It would jagged, going up and down. And the smaller the population, the more jagged it became. Okay, so in a finite population, that beneficial allele probably will still go towards fixation. Whether it reaches it or not kind of depends on how much it's bouncing around. If it's bouncing around a lot, it might actually get lost in some cases, actually go down to 0, even though it's beneficial, just because of all that bounce that it's doing. So if it's bouncing way up and way down, even if the ultimate trajectory is pushing it towards the ceiling, it may accidentally hit the floor in one of its bounces, and so we lost it. So there it was a good allele, and we just lost it because of that bouncing around. So in that case, drift would have changed the outcome. So let's take another example. What happens if instead of it being a beneficial allele, we actually have heterozygous advantage? Remember, we also called that overdominance. Remember what overdominance was? The heterozygous had the highest fitness. So the two homozygotes have lower fitness, the heterozygotes have higher fitness. And back in chapter 6, when we did models of that and we ran a very large population, basically what happened was the population went to some intermediate value of P and Q. So maybe it's 0.5 and 0.5, 0.6 and 0.4, some intermediate value. It went there and it just stayed there. It stayed there indefinitely. We could have run that simulation a billion times, and it would have time periods, and it would have just kept right at the same equilibrium. That's in an infinite population. However, if we now take that population and we drop the size of it, so now instead of that nice smooth line, it's bouncing around again. And what's likely to happen or could happen is, again, depending on how much it's bouncing, it might actually go to fixation, so it might hit the ceiling, or it might go to it might get lost, which would be hitting the floor, right? So it's bouncing around in the middle, but depending on how much that bounces, you might get fixation or loss. So the size of the population is really going to influence what selection does. Those two things are definitely going to be dependent upon one another. So that's basically what the heart of the neutralist selectionist debate is. We're asking which one's more prominent in evolution. Is it drift or is it selection? So right here, our image of this scale with drift and selection is showing us that every population is going to have sort of a slightly different trade-off here. So drift might be heavier and have more of an influence. Selection might be heavier and have more of an influence. And what that's really going to depend on is two things. How strong is drift, which relates back to how strong, what size is the population, right? So if the population is really small, then drift is going to be really strong. If the population is big, drift is going to be weak, right? And then the selection strength is actually going to depend on the environment and the trait that you're looking at. So that's going to vary just based on other factors. So think of it this way. If you have a large population, so you have a really large population, and it's under this trait in our population is under really strong selection. So which one do we think is going to win? What's the large population? So drift isn't really a concern. And the strength of selection is very strong. So selection is going to win. Selection is going to drive that trait to whatever, probably fixation for that particular, let's say, allele. On the other hand, if we look at the opposite condition, where we have a really small population, so drift is really strong, and with really weak selection, then drift is going to win, right? So what happens in that population is going to be more random. It's not going to really be adapting that particular organism towards a particular environment. So ultimately, it's going to depend on those two factors. So the data from flower beetles, red flower beetles, simulate or show, not simulate, it's empirical, experimental evidence, to look at the playoff between drift and selection. So what you have here is you have three phenotypes. They're represented by three genotypes, which is nice. So in this case, our heterozygous has an intermediate phenotype. Now that doesn't mean this is heterozygous advantage. This is called incomplete dominance, and that's just where your heterozygote has an intermediate phenotype. So technically, it's not the highest. It's not overdominance here, because really this B plus has the highest fitness, and then probably the brown one, and then probably the black one. So the wild allele is red, typically has the highest fitness. Okay, so what happens in this population over time? Well, the answer is depends. We have two populations here that we're going to run over time. And well, actually lots of populations, but each one, we ran a bunch of populations that were size 10 and a bunch of populations that were size 100. These small populations down here, when we run them, what we're seeing is a lot of fluctuation. That's that bounce I was telling you about. And so we see that we know that the plus allele, which is what we're plotting here, is advantageous. And so we are seeing a slight trend of it increasing. And in most of the outcomes, it does get higher and higher. But there is a lot of this bounce. And notice right here at around Gen 7 or so, we actually lost that allele in that population. So here's a population that just lost a very beneficial allele because of drift, because of a small population size. Okay. Now, if we look at the graph on top, what we end up seeing here is this is a larger population size, and we're still seeing sort of the same ultimate outcome, ultimate trajectory with that black line, but we're also seeing the fluctuation around it. Notice it's not as much. The bigger this population would get, the smoother this would get, and eventually at infinite it would probably follow a very, very solid straight line, and it wouldn't go up and down. So what you're seeing then is in this top population, we're getting selection favoring that particular allele, and in all of the eventual outcomes, that allele went to a really high frequency. So here, selection was a stronger force because we had a larger population size, not as much fluctuation, less genetic drift. Down here, drift played a big role in the outcome because of the small population size. Okay, so let's take a look now at going back to this. Let's look at sort of the history of the neutralist selectionist debate. So we're going to get a little bit, not a lot because it's intense, but a little bit of the history. So think of it this way. Early on, evolutionary biologists looked at evolutionary change, and they realized that at the molecular level, when they were looking at all these, protein changes, DNA changes, what they were seeing really didn't match what they would have expected to see if selection was the predominant force. And of course, this was kind of mind-altering for most evolutionary biologists because we tend to think of terms in terms of selection being a big, strong force. And so, but they were seeing the patterns really could not be well explained by selection. So they started thinking, and they said, well, if it's not selection that's explaining sort of this rapid change, this clock-like change in DNA that we often see, what does? What is it that could give us that pattern if it's not selection? And basically they came up with the idea that it could be drift, right? So that's when drift force was thrown out there as a mechanism to be seriously considered, was really in an attempt to try to say, hey, selection isn't really explaining these patterns, so we better go and see what is, and drift was the logical choice there. All right, so what they did then is said, let's go and let's see if we can have data that will kind of guide us into evidence for the neutral theory versus selectionist theory of molecular evolution. So they went in, and now see, if you'll notice here, one thing I want to point out, if you notice here, see the line that we're looking at there? That's that clock-like thing that we're talking about. Remember our forensic EA light simulation, where all we had in that particular simulation was mutation. It was the only thing that was happening. And we compared DNA over time and looked at the number of DNA differences from the beginning to each generation, or I think it was every 500 generations, from the initial time period. And we got a line, and we had an equation for our line. And so what it was showing us is that it was a linear relationship between time and number of substitutions, right? And we didn't have any selection in that model. There's no selection in there. It was really just mutation. So again, that's why evolutionary biologists were looking at saying, this pattern that we're seeing, this linear pattern, really may not be indicative of selection operating here. Okay, so let's take a look at this evidence. Essentially, let's ask a couple of things. If neutral mutation occurs more quickly than non-neutral, then that would of course tell us that the neutralists are right? Because that's their idea, is that most mutations are neutral. So let's look for some data and see if the neutral ones are happening. So this is what this data set is trying to do, is to try to establish, are neutral mutations happening faster than non-neutral mutations? Okay, so look at the data. These are data from influenza virus, and if you look at those data, what you see is the black lines over here, and the top graph has a steep slope. And what that steep slope is telling us is that over time we are accumulating those kinds of mutations pretty quickly. These are synonymous mutations. Remember, synonymous mutations are going to be neutral, right? They have to be neutral. You're bringing in the same amino acid. Well, they don't have to be neutral. We'll get to that later. But for now, they tend to be neutral. Let's put it that way. So they tend to be more neutral because they're bringing in synonyms, and there's more of them. Down here, where we see the non-synonymous mutations with this beige line, that's a much shallower slope. It says they're not happening as rapidly. So these data are consistent with the neutral theory, because it's telling us that these neutral mutations are accumulating and happening much more rapidly than those that actually result in a new amino acid, or a new codon and a new amino acid. Okay, so it appears that we see a much higher rate in the neutral mutations. So that would be consistent with neutral mutation being a stronger force. Now let's go back for a minute and let's look at non-neutral mutations. Now what I mean here is that these are mutations where a replacement occurs, and a replacement is going to be, we're going to bring in a different amino acid. Okay, so we have a mutation, and that mutation does not code for a synonym. That means that another amino acid is coming into our protein chain. Now, just because we bring in another amino acid does not necessarily mean that we're going to have a huge effect on our protein. Depends on where the change is, right? And what kind of an amino acid we traded out, right? So some amino acids are a lot like one another. They're both polar or whatever, and so they act the same, and then other amino acids are real different from one another. So it really depends what the amino acid change was and where on the protein it happened. Did it happen in the active site of an enzyme? Probably not so good. Did it happen in some sort of section of the protein that it's sort of all folded under and isn't really exposed? Then it might actually not make much of a difference. So that's what we're looking at here with these three possible effects. You went in and you changed an amino acid. What happened? So, well, the first one is it was a bad thing, right? We have a harmful mutation, wasn't good, and so selection is going to obviously work against that because that was a bad mutation. The second one there, we changed the amino acid, and the protein actually works better now. Okay, so it's a beneficial replacement, and that would of course be under positive selection. If this is a better enzyme, then we're going to select for it. The third possibility is, even though we changed the amino acid, it's really not having an effect on protein function. So that's not really going to be acted on by selection, but it might change based on drift, right? Because it's sort of a random thing. Okay, So again, if we go back up here and we look at these harmful mutations, the type of selection that we refer to when we're looking at something that's harmful that's negative selection or purifying selection. We're going to weed those out. And then these beneficial replacements are going to be under positive selection because we're going to increase those in frequency and keep them around. And then of course the ones that don't have an effect, they might increase, they might decrease, but that would all be by drift. So the data here show some of the relative rates of synonymous, and in this case when they say synonymous, they aren't really meaning back to the code. They mean that the protein itself, the amino acid substitution was synonymous. Okay, so you basically went in and replaced an amino acid but it was similar or synonymous with the one that was there. Well, actually what they're doing here is looking at the overall effect. So if this protein works the same way, it's synonymous. If the protein works a little differently, it's non-synonymous, right? And what it's telling us is that if we look at this 45-degree line, the dotted line that you see there, what that is telling us is along that line we would have equal rates of harmful and beneficial mutations. So the synonymous and non-synonymous would be equal there. So look at where the data fall. All those data, and you can see the proteins that are being recorded there, those data points. So these are data from humans, mice, and rats for various different proteins, hemoglobins, and some of those different proteins you see there. And if you notice, all of the data points are way below the line and off to the right. So what does that tell us? It tells us we have way higher rates of substitution for synonymous change than we do for non-synonymous. So when we do get changes, they are much more likely to be synonymous. And beneficial mutations, which would be the ones that would be up here, basically, are very rare. So again, these data are very supportive of, well, it could be neutral theory. This also could be telling us that there's a lot of negative selection going on for many of these changes. So the data from slide 13, or I'm sorry, the previous slide, also tell us there's a wide range of evolutionary rates across loci, right? Because if we come back here and we look at all of these data points and we look at the rate of substitution for these different proteins, it's just all over the place, right? So there's a wide range of evolutionary rate across the different loci. So if you were to ask, well, what is the rate of mutation or the rate of evolution? We would say, well, it depends. It depends on the organism, as we saw like with HIV versus bacteria versus humans earlier in the semester. And it also, now we're seeing, depends on the locus that you're looking at. So some loci probably mutate and evolve very rapidly, and others very slowly. So why do we think that is? Well, if you look at it, there's actually a really good reason. Some loci probably code for proteins that have very fundamental basic functions, and slight variations on those probably won't work as well, right? Because these are processes that have been going on for millions of years, have been fine-tuned, and so to find a better enzyme by randomly changing the one that we already have to run glycolysis is probably not likely to happen. So those enzymes that are running some of those very key functions in the cell are probably going to have very, very low replacement rates. Histones, right? Histones are very critical to the functioning of cells and being able to give access to the right places for transcription, etc. And so those probably don't change very rapidly. There's others that might be have very low replacement because their three-dimensional structure is very constrained. Okay, so one reason why they might have low replacement is if they're just very critical, and changing them is often just not going to work. The other is, what if you have a three-dimensional structure that is very, very sensitive to changes? So this enzyme's only going to work if it's shaped exactly the right way, and like a lock and a key, and there's just no way that it's going to work if it's even slightly off. And then if you remember studying enzymes, they talked about this other thing called induced fit. So now in this case, that enzyme is maybe a little more flexible. So one molecule comes up and binds to it, and it kind of shifts and moves a little bit. And if it's possible for that enzyme to be a little more flexible, those kinds of proteins might actually be able to have a higher replacement rate. So if your three-dimensional structure is very tied in and it has to be that way, you're probably going to have a low replacement rate. If not, then you might have slightly higher mutation replacement rates. Okay, so what is the neutral theory really giving us? Like what's the value of the neutral theory? Well, the value of the neutral theory, there's others, but one of the values of the neutral theory is that it's kind of like the Hardy-Weinberg theorem. In that, remember what the Hardy-Weinberg did for us? It wasn't actually that useful in telling us exactly what was going on in a population or describing a real population, because remember, in that Hardy-Weinberg population, you had to have no selection, no migration, no drift, random mating, huge population. And we said, that doesn't really describe very many natural populations, right? Most of them are all over the place. So most of them have at least one or two of those assumptions invalid. But that we didn't throw away the Hardy-Weinberg, because what it did for us is, one of the things it did for us is it gave us a null model. What it did is it said, in the absence of selection, migration, drift, etc., what we expect to see in a population that is not evolving is no changes in allele frequencies. So basically what it did is it gave us a model upon which we could base predictions. Then we could look at our actual, compare them, and then we could run statistical tests to see if our actual and predicted outcomes were the same. That's basically what neutral theory is giving us. But in here, it's giving us the null model for selection. It's saying, this is the pattern you should see in the absence of selection. If there is no selection, here's what you're going to see. So we know what we're going to see without selection. We look at a natural population, see what we get, compare the two, and then we can answer the question, is this population under selection? So that is the real value of the neutral theory as a null hypothesis. Okay. So this next slide can be a little tricky. What we're doing here is we're going in, and imagine that we went through and we just looked at a whole big stretch of genome, and we said, in this genome, I'm going to compare it to some ancient ancestor, and I'm going to look at all these changes, areas where there was a change. It's different now, and then I'm going to further categorize these differences. So remember our EA light simulation, we look for differences. Imagine if we could go back and actually look at codons in those differences and say, this difference led to a different amino acid. That one did not. So we go through and we look at all these changes throughout the genome, and we put them into basically two buckets. This change resulted in a replacement, and this change did not. Okay? And so what we're doing is that's what these DN and DS's are here. The DN signifies a non-synonymous mutation. So it was a mutation that gave us a different codon, gave us a different amino acid, right? And then the D sub S was the synonymous, right? So in the synonymous, we essentially got a replacement that had the same, essentially, protein function, right? So if you're non-synonymous, in the first equation there, top equation, if your non-synonymous is a small number, smaller than the synonymous, then this will be less than 1. And that's going to happen when these non-synonymous are bad, right? If these are bad, the number's going to go down, right? Because they're being eliminated. So these are bad mutations, and they are decreasing, or they're small, and that's why this number is less than 1. In the middle equation, the two are equal. So it doesn't matter whether it's non-synonymous or synonymous. So that's going to happen when your replacements are neutral, right? They have exactly the same consequence, so they're neutral. Your bottom equation is saying if your non-synonymous mutations are advantageous, you're going to have a lot of them because they're good, right? And you're going to see that where if this number is large, DN is large and DS is small, this is going to be greater than 1, and that's going to tell us that those are beneficial mutations. So in essence, we can go in and we can look at this ratio, and we can use this ratio to tell us essentially for particular different loci which of these replacements were harmful, neutral, or advantageous, if we can go back and compare them. So let's take a look at some data. Here's some data from showing the ratio of the synonymous and non-synonymous substitution rates. Again, non-synonymous is on top. For the BRCA1 gene, in essentially we're focusing on the primates, and what you're seeing here is any of these numbers that would be 1 would consider that that's a neutral gene. Whatever the changes were that occurred were neutral. If the number is less than 1, the changes that we see are possibly negative, not good mutations. And then if those numbers are greater than 1, they're probably beneficial mutations. And so what you're seeing here is throughout we see them sort of neutral, sometimes possibly a slight negative selection on a particular mutation, which is something we often see, right? It's usually most mutations are neutral or harmful. But then look at the lineage that led to humans and chimps. For whatever reason, this mutation that we had made it so that that replacement had a positive effect. And this is an indication of positive selection on that particular mutation that occurred there. Okay, so this is if I look at a phylogenetic tree and I do that where again we compared all these DNA sequences and then we asked which ones were replacements, which ones were or synonymous replacements, which ones were non-synonymous. In essence, if I see this number greater than 1, it's showing me that right there is a locus that was under positive selection. So it has some predictive value. It allows us to make some pretty interesting conclusions on some of these data sets that we have. So what other loci are under positive selection? Well, we see quite a few actually, but some of the ones they list here, that top one represents what we call an evolutionary arms race between pathogens and hosts. So if you think about it, and we're unfortunately thinking about it way too much these days, but pathogens evolve resistance, right, to their hosts, and they're under selection to do that. under strong positive selection to overcome our defenses, and then we're under a very strong positive selection to overcome their attack, right? And it's kind of a vicious cycle. It keeps going and going. So that's why they call it an evolutionary arms race. So we know that our immune system and their resistance is under very, very strong positive selection. Another one they mention here is these recently duplicated genes. So you take a recently duplicated gene and then it takes on a new function. And now there's all sorts of positive selection that can act on that. So think about the duplicated gene that we saw in the Duke Langers, that RNase 1 gene that was duplicated. And remember what happened is the old gene continued to break down or function in the sort of more neutral pH, but then the new gene was positively selected to actually start working at a slightly lower pH. And that gave individuals that had that particular copy an advantage because now they could eat a new food source. So when you get a new gene, it can very quickly go under positive selection by taking on new functions. And then they list a whole bunch of other ones here that also have been associated with positive selection. So let's look at a slightly different concept. What about a silent mutation that might actually have selection working on it? That sounds a little bit like an oxymoron, actually, because silent is usually telling us that it has no effect, right? And that's why silent is in quotation marks there. So what's happening here is we have a mutation that is silent. So let's just imagine it codes for the same exact amino acid, right? So it's a synonym. However, selection is actually working on it. And we'll see that selection can work on it either directly or indirectly. The direct selection we're going to look at as codon bias, and then we'll see a couple of ways that we can see this indirect selection through hitchhiking and background selection. So let's take a look at that first one. Here you have a mutation. You get the same exact amino acid sequence, but it's still being affected by natural selection. And so one of the ways that can happen, this isn't the only way, but one of the ways it can happen is through codon bias. So in the past, when we looked at changes in DNA and we found that those changes brought in a synonymous amino acid, we automatically assumed that they were neutral with respect to fitness. I mean, it's the same amino acid, so it's going to be the same fitness. What if that isn't true? So if it's true, we can make a prediction. If it's really neutral, then we should use all the synonyms in the proportion that they are represented in the genetic code. So if you go in and you look at, for instance, if you look at the genetic code and you say, here's amino acid X, and amino acid X has 2 different codons that go with it, 2 different 3-letter words. So when I'm building a protein, every time I want amino acid X, 50% of the time I should be using one of those codons, and 50% of the time I should be using the other one. That would be random. So I just flip a coin, and then I pick which one of those codons I use. If that's not the case, which we know it's not, if it's non-random, then that's what we call codon bias. So if you're not using those codons in the same proportion that they exist, it's codon bias. So there's a couple things we know about codon bias. One is that it is very strong in highly expressed genes. So those genes that we express a lot of, we transcribe a lot and then translate a lot, it is very high. It's very weak, or sometimes maybe even non-existent in genes that we rarely express. So we do know that there's a bias there. And another thing that happens is that when we do have the bias, the codons that we tend to use So if I use, instead of using them 50-50, I use this one codon like 90% of the time instead of the other codon. What I'm finding is that codon that I used, that 90% of the time, is also correlated with the tRNA species that is most abundant in the cell. So remember, you have these two codons that go with this amino acid X. And those two codons are going to match up with two tRNAs that have two different anticodons. And they're going to be attached to their tRNAs. Well, what if you only you have a lot of one kind of tRNA? Well, if I use that codon, that matching codon more, I'm going to be able to build my proteins more effectively and efficiently. So they think that one of the reasons why we have strong codon bias throughout our genome is that it basically favors translational efficiency, right? Because if I use the one that I have the most of, when I reach out, so imagine your cell is your ribosomes, you're making proteins, and they're randomly reaching out to grab, okay, I need alanine right now. And it reaches out and it grabs a tRNA that has alanine, but it doesn't match the codon that is sitting there. So it has to throw that one back and grab another one. Well, if the one it's looking for is really rare, it's going to have to do that a bunch of times. Nope, that's not the right one. Nope, that's not the right one, until it gets the right one. If instead we have one that's really common and we use that one, more often than not, when I reach out down into this pool and I pick up one of those tRNAs, it's going to be a match, right? And so that's going to make translation of these proteins much faster and probably more accurate as well. And so selection is going to favor that. So that's direct selection, acting on efficiency, which would then generate bias. So here is some data on codon bias. And one of the things that we see is, so we see evidence here of codon bias for two different codons. These are codons that are going to, or I should say two different amino acids. We have leucine and we have valine. And if you look at the top, and we have three different organisms: bacteria, yeast, and fruit fly. And so what you're going to see is for leucine, there are 6 different codons. So if we used them all in equal proportions, then we should have, those bars would have 6 evenly spaced little boxes inside, right? And we kind of do see that more or less for the bottom ones here. And those are lightly transcribed genes. So these are genes we don't use a whole lot. Here, the heavily transcribed genes that we use a lot, we see a very strong bias. I mean, almost exclusively in the bacteria, we use the CUG for leucine. We don't use the other ones. We see it a lot in fruit flies as well. Yeast has a bias. It just happens to be a different bias, but they still have a bias. And then the bottom two are just looking for exactly the same thing, but for valine. And again, we see much more randomness and much more even use of codons here in the lightly transcribed genes, and much more bias up here in the heavily transcribed genes. So much more bias in our heavily transcribed genes, which makes sense because those are the ones that we need to be efficient with, right? These data here are showing is this is trying to draw now the correlation between the codon bias and the availability of tRNA. So if you look here on the left side, we have E. coli, and the first graph here is showing here's the frequency of all the different tRNA, or the tRNA that is attached to this codon or anticodon, right? And then this is the one that's attached to this other codon, etc. So what we're finding then is the most abundant tRNA that we have in the cell matches this particular codon, and then we look at which one is used the most, and in the highly expressed genes, there it is. So these match. So the more tRNA we have, that's the codon we use. That's the codon bias in highly expressed genes. Not the same in rarely expressed genes. So this is a lot more even. So this draws that correlation and says this bias that we see is probably due to the fact that we have different amounts of these different tRNAs available in the cell. that was looking at the direct effects of possible selection working on synonyms, even though we thought that they shouldn't. We said, wait a minute, these are silent mutations and selection's still working. Now we want to look at some indirect effects. So now instead of selection actually working on, let's say, that particular locus, that particular codon or whatever, now what's happening is here we have a silent mutation and it's sitting there. And there's two possible things going on in this slide. That neutral mutation is sitting next to a good gene, or maybe it's sitting next to a bad gene. The sitting next to a good gene is the first thing described there, the hitchhiking. Here we have a neutral gene. It could even have a slight harmful effect. And it's sitting there, but it's sitting there next to an awesome gene. And that good gene is under positive selection. And it will increase in frequency. And guess what? This neutral gene, or maybe even slightly bad gene that's sitting right next to it, is going to go along for the ride because they are tightly linked. And so selection on one locus is causing the increase in the other locus. Opposite is happening in background selection. Now that neutral gene or allele mutation sitting next to a section that is under negative selection. It's being removed from the population because it has a harmful consequence. And so even though this mutation that is sitting here is neutral, it's being thrown out sort of with the bathwater because the genes next to it are being eliminated. And so that's what we refer to as background selection. So this next slide leaves us to a sort of an unfortunate conclusion. We find this a lot in biology, that we look at lots of different theories and then we come to the end and realize that we don't really have a really great answer as to which one is more prominent, or can we get rid of one idea and just stick with the other one. And right now it still remains fairly undecided. There are some patterns that we see that look like they are neutral, and then there are a lot of other patterns that we see that look like there has been a lot of positive selection acting on different loci. So I think that the bottom line is that we shouldn't really expect a one-size-fits-all answer here. That I think the answer is going to be it depends. It depends on the organism, it depends on the circumstances, it depends on the loci involved. And, of course, when you're looking for big patterns in life, that's a fairly unsatisfying answer, because what it means is that there aren't any really strong general patterns. To end this particular session, what I want to do is just point out that on your PowerPoint and your notes and everything, it tells you to omit the section from the textbook on coalescence. So at this point in the textbook, you'll see a fairly significant little section there on going into this concept of coalescence, and we are completely eliminating that. So I will not ask you anything about coalescence. You could just skip right over that part. And I will return soon with the second part of this lecture.